Hi, everyone. I am Becky Chong, and I am the Awards Director for the Systematic Society of Systematic Biologists. And it's my pleasure to bring everyone back for the second and final session of our Mayr Symposium Award. And again, this is considered SSB's premier award, recognizing the incredible work and contributions to the field of systematics that some of our early career scientists have been able to accomplish over the period of their PhD. And so with that, I think we're ready to go. Thank you.
Yeah. So should I start the presentation?
Yes, go ahead.
Thank you. I hope I'm audible and my slides are visible. Hi, everyone. Greetings to everyone who are attending from different time zones of the world. I'm Tania, a second-year PhD student at the Department of Life Sciences from Shiv Nadar Institution of Eminence, India. I'm glad to be a part of Evolution Meetings 2025, and I thank SSB for giving me an opportunity to present my work in this Ernst Mayr Symposium this year. So let me introduce the unique and enigmatic subterranean fish that is shown in this picture, Pangiobugia, that inhabits the lateritic aquifers of southern India. Today, I'll be sharing about the origin and evolution of subterranean eel loaches of southern India. Pangio, commonly called as eel loaches, are very small-sized freshwater fishes belonging to the family Cobitidae. They are very small, eel-like anguilliform cypriniformes that are generally bottom dwellers, mostly burying themselves in sand or gravel in the stream bottom, or maybe in leaf litter and dense aquatic vegetation. In general, cobitids are found across Eurasia and parts of Morocco. This specific genus Pangio is distributed across the tropical streams of South and Southeast Asia. So as you can see in the map, the Sunda Islands harbor the greatest diversity of Pangio with 22 valid species. Also, there are 34 valid species of the genus Pangio distributed in these geographical regions, the type localities of which are plotted in the map. These are also very popular aquarium fishes because of their vivid coloration, banding patterns, their peculiar behavior, and all. So you can see an overall view of these fishes and the geographical areas that they occur in the map. In contrast to the high species number in Southeast Asia, only a few eelote species are known from India and its adjacent areas. So our particular focus is on the species that are occurring in India. The species that are known from India are the Pangea from Northeast India, Pangiogoensis, Pangioammophila, Pangiobugia, Pangiojuhue, and Pangiopathala from the southwestern part of India. So it is clearly evident that there is a disjunct distribution of these species within India, with no species of Pangeo recorded from the central part of India. So coming to our next question, why the southwestern part of India is interesting, especially in the case of freshwater fishes? Because it is where the hottest hotspot of biological diversity is present, the Western Ghats and the Sri Lanka biodiversity hotspot. It is a region with exceptionally high levels of species richness and endemism. This mountain range extending along India's west coast harbors a significant portion of the country's flora and fauna, including many endemic species. It is also recognized as one of the world's most important areas for conservation. We have 5 species of eel loaches distributed along the region of Western Ghats, with 2 species occurring in the surface waters and 3 species occurring in the subterranean aquifers. Even though they all belong to the same genus Pangeo, we can clearly see differences in their morphology. What could be the reason for these differences? Obviously, it is the difference in the habitat or the ecosystem that they are thriving in. So we have 2 different types of habitats over here. While the surface forms of Pangeo inhabit shallow, sandy areas or streams with dense aquatic vegetation and leaf litter, the subterranean forms of Pangeo inhabit the narrow channels in the lateritic aquifers that are present along the mid-elevation coastline of Western Ghats. So you can clearly see a difference in the habitat of these organisms and how their morphology could have developed or could have modified because of that. So these subterranean lateritic aquifers are unique ecosystems that host a wide assemblage of various subterranean fishes, not just Pangeo. They're similar to a cave ecosystem, as you're familiar with, where there is complete absence of light, lack of oxygen, and other nutrients as well. So we can see that they are porous rocks that are generally forms the base of a homestead well. This is the homestead well that is shown in the figure. It would be connected to a nearby aquifer. So there is a constant movement or migration of these subterranean fishes between the aquifer and well through these narrow lateritic aquifer channels. So in order to traverse through these narrow pathways, they must have developed a bunch of morphological, physiological, sensory, and behavioral adaptations. So as we can see in the picture, these fishes have reduced eyes, the subterranean ones, have reduced eyes, enlarged sensory barbels, and they lack dorsal and pelvic fins. These adaptations might be helping them to thrive in these extreme environments. So one might ask, how are these fish in the same genus when they look entirely different from each other? Are they same? Are they different? Do they have any relation with each other? The answer is we don't know. Sometimes when the morphology is very deceiving, the genetic information is where we rely upon. With these information from the existing literature, there are some open questions that are in front of us. What is the relationship between Western Ghats and Southeast Asian eel loaches? What is the reason for the distant distribution of eel loaches in Northeast India and the Western Ghats? How and when the origin and diversification of surface and subterranean forms of eel loaches in Western Ghats happens? So these are the questions that are open in front of us. To address these questions, we have formulated 3 objectives. Understand the phylogenetic relationship between Southeast Asian eel loaches and Western Ghat eel loaches. Understand the origin, evolution, and molecular systematic of the eel loaches of Western Ghats. And understand the biogeography of eel loaches using time-calibrated phylogenetic tree. So these are the methods that we have followed. The sampling of specimens were conducted along the Western Ghats for the genus Pangeo as well as other cobitids available. Also for the subterranean fish sample collection, it was through a citizen science initiative because these ecosystems are not accessible directly to humans. Then the DNA was extracted from these fish specimens. And the gene of interest that we have are 3 genes, the cytochrome oxidase 1, cytochrome B, and the nuclear gene RAG1. These genes were amplified using PCR, and they were further sequenced by using Sanger sequencing. The quality of the sequence were checked, and the alignment was done in MEGA. Additional sequences of cobitids, of all the cobitids available, were retrieved from NCBI GenBank, and all this data was compiled for further analysis. So this genetic data was subjected to different analysis in different tools. So first was the molecular phylogeny done in IQ-tree by the maximum likelihood method. Second is the time tree construction by using rel time. We used 6 fossil records of Cypriniformis as calibration points. And this data was then again subjected for biogeographic analysis in RASP during using the dispersal extinction cladogenesis model. So coming to the results, we can see a very beautiful phylogenetic tree over here. So this is the first comprehensive phylogeny for eel loaches from southern India. So as you can see here, there are 3 clades of Pangeo that I have highlighted over here. The first clade is Pangeo sensu stricto, that is the ancestral lineage of Pangeo. Clade 2 is the Pangeo shelfordi group that is coming closer to the Pangeo sensu stricto. And third is the most interesting and is our main focus, that is the Pangeo from the Western Ghats that forms the clade 3. So as you can see here, there is a the Pangeo from the Western Ghats is not actually clading with the ancestral Pangeo lineage, and it is not coming with the Pangeo sensu stricto group. So the southern Indian loaches do not clade with Pangeo sensu stricto, and we suppose that it needs a new generic identity, but only after confirming with further analysis also. But we suggest that it should have a new generic identity since they don't clade with the Pangiosensu stricto, because there are two other genus that are coming in between and that are more closely related to Pangeo in the Western Ghats. And also to note that the southern Indian subterranean and surface forms are sister taxa to each other. So we were asking about the relationship between subterranean and surface fishes. So these are sister taxa to each other. About the molecular systematics of the Western Ghat eel loaches. So there are a lot of taxonomic confusions, taxonomic aberrations in this taxonomy of Pangeo. So as we can see here, the Pangeo Ammophila and Pangeo govensis, they fall into the same clade and cannot be considered as two different species. And they show very little genetic and even morphological difference. And there is a scope for synonymization of these two species. Also, coming to the subterranean fishes, the genetically confirmed Pangeo showed presence and absence of dorsal fin. So recently, this Pangeo with dorsal fin was described as a new species called Pangeo juhuwe. But in the phylogenetic tree, we can see that the Pangeo juhuwe with dorsal fin and the Pangeo that are with dorsal fin, they are indicated in the bracket D. So they are all falling into a single clade and they don't belong to a different species. So they all are same species. It is just that the absence or presence of dorsal fin is a secondary character loss due to the subterranean adaptation. As a result, this Pangeo juhuwe could be a junior synonym of Pangeo bujia. Also, there is an undescribed species of Pangeo from the surface waters. So this could be further examined to describe as a new species. In the biogeographical analysis, it is very interesting to note that the eel loaches originated in the Southeast Asia in the Eocene about 42 million years ago. And the ancestral eel loaches must have probably dispersed into the northern India from Southeast Asia during the Oligocene about 32 million years ago. And this dispersal could have happened through the Greater India connectivity with Eurasia. So while the Indian subcontinent was moving towards Eurasia and while colliding with Eurasia, these species must have traveled through the northeastern India and traveled to in India and then diverged over here. And this is the most interesting part, that is the subterranean origin of the subterranean fishes of the lateritic aquifers of Kerala. So the southern Indian eel loaches, it is shown that it is probably originated in the late Oligocene around 27 million years ago. The common ancestor of surface and subterranean eel loaches separated in the early Miocene about 21 million years ago. This is where it is significant. So we suspect that there was a Miocene drought that happened in southern India. Probably this could have pushed the surface water Pangeo or the eel loaches of southern India, as they cannot be called as Pangeo right now. So the Miocene drought must have put a pressure on them, and in search of survival, they must have gone into the subterranean channels and thrived over there and developed all these adaptations over time. So to conclude my work, the genus that is classified as Pangeo, the eel loaches, they are a polyphyletic group with 3 genetically distinct lineages. So the southern Indian eel loaches that do not clade with the Pangeo ancestral lineage require a new generic identity. Apart from the new generic placement, all the southern Indian loaches need several taxonomic revisions, including the synonymization of 2 surface water fishes and 2 subterranean water fishes and the description of a new species. Also, the biogeographical analysis suggests that the Pangeo in southern India originated in the Southeast Asia in late Eocene and dispersed into India in the Oligocene, probably through the Greater Indian connection with Eurasia. So the southern Indian eel loaches probably originated in the late Oligocene, and we suspect that the Miocene drought must have pushed or must have resulted in the evolution of these subterranean forms. So this is the conclusion of my work, but this is just an initial analysis that we have got, but to further validate all these assumptions or all these results that we got, we need further examination, especially through genomic studies. And to understand the adaptations and all, we need genomic studies. So I would like to acknowledge my PhD supervisor and my collaborator for all the help that they have done for me, and all the people who have helped me in the field and the lab. I'm happy to take any questions. So this is just a typical behavior of Pangeo hiding in the crevices. So yeah.
So because of our timing, I'm going to actually have to direct you to answer questions in the Q&A. There's one already there, but we're at the point where we need you to stop sharing your screen so our next presenter can share theirs.
Sure. Thank you
Because
so
they will
much.
be starting in 15 seconds.
Okay. Thank you. Thank you so much.
All right, so Laura, you can go ahead and share your screen.
Okay, how's that?
And you can begin whenever you are ready.
Okay, thank you. So today I'm going to present some of my research on how we can use model adequacy to determine the fit of morphological models. And before I get started, I just want to highlight my co-authors who were extremely helpful throughout this project. And just to say that, yeah, the work has been published earlier in the year in SysBio, if anyone is interested in that. So morphological data is an extremely important source of information for studying evolution in deep time. And it's the only data that we have available for extinct taxa. I mean, of course, there's some like aDNA, but most of the time we only have what is in the fossil record. So it will be like extinct, and these extinct taxa account for the majority of life on Earth. So it's quite a lot of, evolutionary history that we can only use morphological data to understand. And we can collect morphological data both from extinct and extant taxa. And in this way, it allows us to basically study like how these organisms, study them together and understand their evolutionary relationships and evolutionary histories. Like, for example, we can ask questions about like, when did the octopus lose these shells that we find in the fossil record? So in phylogenetics, as we know, we have these like substitution or character models that are responsible for describing the evolution of this character data. And I have molecular data on this slide. As many of the models that were originally developed, that we use, were originally developed for molecular data, and then kind of secondarily applied to morphology by generalizing some of the assumptions. And it's important to ask, like, does this always make sense? Is this like, are these models doing a good job? Because morphological data is, it's quite complex and it's a lot messier, I suppose, than molecular data. So if we take this like subset of a matrix, it's basically a Cambrian echinoderms. And while it's quite small, there's really like a lot going on. And we have these different traits, here describing the presence and absence of one trait, here the presence and absence of another. So like straight away we can see it's quite different from molecular data in that characters have no consistent biological meaning across the matrix. We also have like traits with different number of states. We have these like special characters. And then if you're using paleo data, you're going to get like a high proportion of missing data. And we start to see these taphonomic processes which remove data from remove data from the fossil record, and it's going to impact our ability to character code. So making any kind of general assumption about how this data was generated, how it evolves, is extremely difficult. And kind of as a result of this, there's been like a lot of debate about whether or not we should be using models at all, and if it's appropriate for this type of data. And this conversation has like dominated especially in like paleontology. It's quite, it's been quite a big topic of conversation. And, it's kind of also prevented us from taking like a proper look at the models that are available and kind of think about how good are they as opposed to comparing them, which is what we tried to do with this study. And basically we wanted to know like, well, first of all, like how can we choose a morphological model? Like how can we say whether or not it's doing a good job? And then secondary, like given that we can confidently identify a model, can we ask are current models fit for empirical data? Do this one first. So just briefly in terms of morphological models, we sort of have one model that we pretty much rely on, which is this MK model. And it's a generalization of the Jukes-Cantor model. So it makes quite these simplistic assumptions, basically assumes that we have equal transition probabilities between states and equal stationary frequencies. And we can relax these assumptions and extend on this model in different ways, given what we've learned from empirical data. And in this talk, I'm just going to, mainly focus on the M1 character rate variation, this like adding gamma. And then we also looked at the MKV model, which accounts for the fact that for morphological data, it's quite common to have all states varying, another characteristic which is quite different than molecular data. And also looked at the impact of partitioning the data. So in the interest of time, I'm not going to show, but basically we first explored how these different these different model extensions impact inference results. And we found that for around 200 data sets, that the results, when we did the analysis under different models, they impacted both the branch lengths and topology. So kind of, emphasized the importance of model choice. But then we were thinking like, what's the best way to do this? So when we think about model choice, there's kind of two avenues, I suppose, when you try to pick a model. And the most common sort of like gold standard would be model selection. And this works by taking multiple models and determining which fits your data the best. So like providing a relative fit. But this in no way provides any justification as to whether or not the model chosen is a good fit, just that it fits better than others. An alternative approach, however, would be model adequacy. And this allows you to determine the absolute fit of a model given a data set. And model adequacy approaches have been applied a number of times in molecular data, somewhat a bit more sporadically in morphological. So here we basically wanted to explore the use of model adequacy for morphological data and develop a pipeline for doing this. And this would just like also allow us to kind of be more confident about the models we're using and say like model-based, approaches are appropriate and say quite clearly if they're doing a good job at capturing these underlying dynamics of our data. So first I'm going to talk about model adequacy. And one approach is to use posterior predictive simulations. And this is, the general workflow. Basically, you have your empirical data, you carry out an inference under your model of interest, let's say MK gamma model. And then during your MCMC, you sample values from your posterior, such as branch lengths, topologies, your alpha values. And then in the next step, you basically simulate data, you simulate new data sets using the parameters you sampled, like you simulate under the same model, and then you carry out an inference. And the main idea here is basically that if your model is really capturing the dynamics of your data, the simulated data should be very similar to your empirical data, which then leads to the question, like, how do we measure similarity? And to do this, we need test statistics. So we need to validate a number of test statistics that allow us to determine whether or not a model is adequate. And as I mentioned, this has been done for molecular data, but not for morphological. So we need to come up with, a number of different ways of measuring it. And these are the ones we chose. We have data-based, inference-based, and mixed. But of course, in order to do this, we needed to carry out a simulation study. And this was kind of the general setup. We chose 2 empirical data sets to base our simulation on. And for each simulated data set, we simulated data under 4 different models with 20 replicates each. And the idea was we carried out posterior predictive simulations under 7 models, one of which was correct, and then looked at the different test statistics to see which could tell us which was the correct model and which could identify incorrect models. So just to show you some of the results of what an analysis would look like. Here you have the results of consistency index. So you have this histogram here, which shows you your simulated values. And during this, we found that 500 was, enough to kind of use. And then this blue line is your empirical. Basically, if your model is doing a good job, you would expect these lines to be very close or, not significantly different from each other. And there are different ways to kind of present this information. So using p-values, or what I'll focus on for the remainder of the talk is using effect sizes. So these plots are essentially showing the same thing, just we've transformed them into effect sizes. And essentially, the closer the value is to 0, the more similar your simulated data is to your empirical. So as I said, we did the simulation study using 7 different models, and the colored one is the correct model, and the 6 different dots would be the different models. And we did this for 20 replicates. So if you look here, this is basically the results from consistency index when we did a simulation under an MK model and when we did a simulation under a more complex model. So we can see the effect sizes are quite small because we did, inference under that. So we'd expect them to be small because we simulated on the model, used posterior predictive simulation under the same model. So we'd expect them to be small. And then when we, did posterior predictive simulations under different models, we can see the effect size get a lot larger because the models, they're misspecified. Especially these results here were extremely nice in that we can see the effect sizes are getting extremely large as we kind of use a model that is less specific, or using the incorrect model, we're getting like these increases in effect sizes. So not only can we identify a model that is correct, we can also state when a model like does not fit our data at all. So this is quite a promising result. And just to save time again, basically we only found that these mixed test statistics, consistency index and retention index, were indicative of this. Anytime we looked at database or inference-based, they weren't informative on either the correct model or the incorrect model. But this was quite nice because this step of posterior predictive simulations is the most expensive because basically you'd have to do inference like 500 times. So, by cutting this out, it makes this approach really, really fast. Basically, you just need to run this analysis. Simulation is usually in like seconds. So it really cuts down the time by not having to do this, which is quite nice. So basically from this, we were like, okay, we can use model adequacy for determining a data set. But we also just want to take a quick look at model selection because, this is like the gold standard, I suppose, for determining a model. And while it's used a lot, it's not really been systematically explored for morphological data. And when we did this, we kind of found this quite weird pattern, wherein if we simulate data under an unpartitioned model, model selection always chose a partition model. So here, for example, we simulated under this unpartitioned model, but every time we were getting support for one that was partitioned. And this pattern was consistent across data sets, across models. And yeah, it kind of came a bit clear when we thought about the way we actually partition our data, and it's how it's quite different than molecular. So in an unpartitioned data set, everything is in a Q matrix of size 4, like here. But then if we partition our model, you know, we're splitting everything up into things that are the correct Q matrix size. And this is impacting the likelihood calculation. So as you know, Bayes factors works by comparing marginal likelihoods. But when we're changing this K value in the likelihood is just going to decrease. So basically, this is, you know, a binary trait. But if you say K is 2, and then if you had this in a Q matrix of size 3, and K is 3, basically the likelihood would just decrease. And just to kind of show that, we took a 2-state data set and calculated the likelihood for like increasing K values, and showed that, yeah, like these are just different branch lengths. So basically the log likelihood was decreasing as we increased the state size. And yeah, while this makes sense a lot of the time, it's just important to kind of think about it's not actually a biological signal. So if you're choosing between different partitioning schemes, you know, you're going to get this result. Basically, you can predict it before you do it. And also it can run into an issue if you're like using data that's like a subset of a larger data set and you don't have all the states in there. That, yeah, model selection it won't give you a biological signal. It'll just basically tell you this. So yeah, just something to like keep in mind, bear in mind when you're using this kind of approach for comparing between partitioning schemes. But yeah, so kind of the first objective we want to see is how do we choose a model? And we found that using posterior predictive simulations, we were able to develop a pipeline and determine consistency index and retention index were informative as a test statistic. And that when you're using model selection approaches that rely on Bayes factors, these can produce misleading results if you want to compare different Q matrix sizes. But yeah, my second thing that I want to look at was, yeah, now that we have a way of confidently determining a model, are any of them actually doing a good job for empirical data sets? So this is just one data set to show you. It has 12 taxa, 51 characters, and 4 states. And again, I'm just going to show you the effect sizes. So this is basically for the MK model. You can see it's not, ideally you want the minimum maximum limits to be like crossing that 0 line. Then we can show that, yeah, it's like adequate. So for the MK model, we can say, yeah, it's not looking good. But as we use more complex models, they're looking more similar. So essentially that's saying the simulated data is more similar to our empirical. So we can show quite nicely that for this data set, we can use any of these models, and it is doing a good job of basically capturing the dynamics of our data. But, you know, for this data set, for example, we didn't find any models to be adequate. They were all kind of, yeah, all the effect sizes were far too big in that the data that we were simulating was quite different from our empirical data. I suppose this isn't like overly surprising given the complexity of morphological data. And I do also want to stress that like the models I explored here, it's not like an exhaustive list. I basically just chose 7 models, 7 kind of combinations that I explained at the start. And yeah, did analysis, did inference with them. But you can obviously, yeah, if you know more about the data, you can kind of explore different versions. But this kind of advocates that, you know, more like while the models are doing a good job, like there's more kind of model development required to, yeah, use all this data to the best of our ability. But yeah, we did find that, you know, obviously I just showed 2 examples here, but in the paper we obviously tested a lot more. And yeah, we found that for a lot of models, a number of empirical data sets for a number of empirical data sets, their models were available. But there's no clear link really between the characteristics of the data set and the model adequacy. And as I said, yeah, further work kind of needs to be done on this model development. And yeah, basically I just want to say that, you know, using posterior predictive simulations, we can really kind of like with absolute, like talk about like the absolute fit of a model, which is quite important for morphological data given its complexity. And given that we're taking models from molecular data, we can use this approach to say for sure this model fits and kind of remove the need for that like ranking system. And while we do often find models available, further work is required. And just to kind of emphasize that, yeah, this workflow can be applied to any model of discrete character evolution. And this is the paper here, and there's a QR code for a tutorial for this in RevBase if anybody is interested. And thank you.
So thank you. I'm sorry, unfortunately, we're not going to have time for questions because it's the next speaker is speaking in 30 seconds. So we're going to have you stop sharing your screen. We'll get the slides for the next speaker up. And as always, I'm directing if you have questions for a previous speaker, feel free to put them in the Q&A and people will be able to answer questions there. That being said, Sarthak, we're going to have you wait about 10 seconds and then you can begin speaking.
Can I speak now?
Sure, go ahead at this point.
Sure. Hello, everyone. I'm Sarthak. I'm a PhD student at Indiana University Bloomington, and welcome to my talk. So let's go. Next slide. Give me a second. We have some technical difficulties. Okay, so let's begin with the gene tree in the middle of the screen. There's a species tree shown in black, and I want you to take a moment and think, what is the evolutionary history of this gene tree? With this question, I'm asking, how many duplications occurred in the history of the gene tree? Were there any losses? Did the duplication happen before speciation or after speciation? Or which gene trees are paralogs and which are orthologs? Next, one more. All right. So you probably think like this history with one duplication on the top and loss at B is the most probable history of this gene tree. But one slide more, sorry. But this history with one duplication can also be the history of this gene tree. Both of these history can significantly change our understanding of this gene tree. And to answer this question, we need to see the interaction of duplication, losses, and coalescence. What I want to convince you by the end of today's talk is like the bottom tree is more likely given some history of duplication and coalescence. To answer this, we need a unified model of duplication, loss, and coalescence, SIFT. Next slide. All right. So what does a unified model of duplication, loss, and coalescence look like? And then once we know that, we can ask the question, how many trees are possible with one duplication? Next, do all trees occur with equal probabilities? And finally, can we recover the true evolutionary history of the gene tree? Okay. So there's a unified model called multi-locus multi-species coalescent called MLMSC, introduced by Lee et al. In 2021. This model unifies duplication, losses, and coalescence under one framework. On the screen, you see two loci, parent locus and daughter locus. The blue box and cross represents the sequence at these loci. In MLMSC, one more, sorry. In MLMSC, under multi-species coalescence, there are both parents and daughter locus trees are drawn from multi-species coalescence, and the parent tree is the parent tree is concordant to the species tree, whereas the daughter tree is discordant to the species tree. Next, when the duplication occurs, given by the red star, genetic material is copied from one locus to another. For example, the blue sequence is transferred to the daughter locus. The duplicated sequence thus follows the gene tree at the daughter locus. Next, two more. Okay, so duplicated sequence also has to also carries the history of the parent locus, so it has to coalesce back to the parent locus. So when we are modeling the unified model, we are modeling three coalescent processes: one at the parent locus, one at the daughter locus, and one at the one coalescent process between the parent and daughter locus. Next, next slide. All right, so. Here, give me a second. Here we dive deeper into the coalescence between the parent and daughter locus. The duplicate can coalesce with any branches in the parent lineage as long as it's above the duplication time. Let's walk through an example of what that might look like. So here you have a duplication NC. It can coalesce with C in the parent locus, and that's the most probable tree because it has more time to coalesce with C. And the next one, if it fails to coalesce, it can coalesce with B, or it can coalesce with BC. You can coalesce with A, or it can coalesce with ABC. So with one duplication, you have 5 branches where it can coalesce. Okay. Next slide. All right. A natural question to ask now is what happens if the loci are linked? If the loci are unlinked, the gene trees evolve independently. But if the loci are linked, the trees may look identical, but the mechanism of duplication and coalescence still remains the same. So linkage affects the tree shape, but not the underlying evolutionary process. So 2 more. Yeah, next slide. Okay, so now let's summarize ML-MSC model. So in ML-MSC, each locus evolves along its own gene tree, right? The duplication moves sequence from parent to daughter locus. The duplicated sequence follow the gene tree as a daughter locus, and all parent and daughter locus must coalesce with each other. Now that we have a model of duplication and loss and coalescence, now we can ask some question, like what question can we ask with it? So next slide. The first question we can ask is how large does the gene tree space become, like with just one duplication, right? To answer this question, you have to answer how many places can the duplicate fall on in the daughter lineage and how many places can it coalesce back in the parent lineage, okay? So, right, let's begin with the simple case of 2 species, A and B. So we'll ask how many places can a duplication occur. So here are 3 places it can occur. It can occur in A2, B2, or A2, B2, which are just A, B's in the daughter locus. Okay, but then, so let's pick one of those duplicates and see where it can coalesce back in the parent locus. Okay, so the daughter, so we picked the A lineage. So next slide, and then it can coalesce with A1. A in the parent locus. It can also coalesce with B lineage in the parent locus, and it can also coalesce with AB lineage in the parent locus. So for one duplication, you have 3 places to coalesce. Great. Next slide. So next. So you have 3 places in which the duplicate can occur in the daughter locus, and you have 3 places where it can coalesce back in the parent locus. So you have like 9 total trees at the end of the slide. Okay. So now let's see what happens if we go from 2 to 3 species. With 3 species, there are 9 possible insertions. 9 possible places where the duplication can occur, and there are 15 places where the duplicate can coalesce back in the parent locus. So you have 135 different trees. So here's one such example. The duplication is happening in C in the daughter, and it coalesces with A in the parent, giving this gene tree. Okay. Next slide. Now let's generalize it. This is the equation that uses the number of species n to count the number of gene trees conditioned on one duplication. So this grows very fast. One more, yes. For 2 species, you have 9 trees. For 3 species, you have 135. For 6 species, you have millions of trees. This shows even with a simple case of one duplication, the coalescence, the number of trees grows like very, very fast. Okay. The next slide. What about if when we add losses into the mix? Here's a gene tree that cannot be generated by one duplication and coalescence, but it can be generated with one duplication, one loss, and coalescence. So how many of these are we really talking about? Next slide. So here's the equation that counts the extra number of gene trees that are possible with one duplication and one loss and coalescence, but not with one duplication and coalescence. For 2 species, you have 19 trees. One more, yeah. 19 trees, which is 10 more than with one duplication. For 3 species, you have 255, which is 120 more than one duplication. When for 6 species, you have millions of more trees. So we see here, like by adding one extra event loss to duplication and coalescence, we are making millions of many trees. So, but of course, all these trees are not equally probable. Next slide. So this brings us to our second question. Are all trees equally likely under ML-MSC? Let's explore this using our exemplar tree. I'm going to use this tree along with time of duplication and time of speciation to show a way to derive the probability expression for the trees. Okay, next slide. So for our exemplar tree, there are 2 possible paths conditioned on one duplication. One path is if the duplication happened before speciation, given by the red star. One more. Red star. When there is a duplication, it must land on A lineage in the daughter locus. That happens with that probability. And when that happens, it must coalesce with AB lineage in the parent locus, giving the gene tree with that probability. One more. So that's the total probability of seeing this gene tree conditioned on one duplication and duplication being before speciation. There's another path when the duplication happens after speciation, right? So this happens with this probability. Yeah, quickly you can go around. Sorry. And then when it must fall on A lineage, and then when that happens, it must coalesce with AB to get the exemplar tree with that probability. So that's the total probability of seeing it. We can use the same technique, next slide. We can use the same technique to find the probability expression for all 9 topologies. This gives us family of curves for different histories based on a time of duplication. Yes. So here are there were 9 topologies for 2 species, but we have 5 curves. This is because some of the trees are symmetrical and have same probability expression. So the x-axis of this curve is time of duplication. The y-axis is the probability of generating the tree. The red vertical line is the time of speciation. The key insights from this slide is if the duplication occurs around the time of speciation, it's hard to determine which tree will be generated because there are multiple trees with like similar probabilities around the time of speciation. But if it occurs before the time of speciation, we tend to get the green trees. One more. This is because if the duplication happens closer to the tip, for instance, at A in the daughter lineage, then it has more chances of coalescing with A in the parent. A symmetrical case of this is when the duplication happens at B in the daughter and coalesces with B in the parent. Right? But then if duplication happens long after the speciation, then we get the blue tree where both lineages coalesce far above the time of duplication. This is because when the duplication happens after the speciation, both the parent and daughter lineages are expected to coalesce. So you don't really land the duplication on like single lineage like A and B. You will land it on the coalesce like ancestral population of AB or something like that. Okay. So next slide. So for 3 species, I won't show you all 35 expressions, 135 expressions, sorry, but similar to 2 species, we can compute them based on time of duplication and time of speciation. But there are 2 time of speciation, so we need to take both time of speciation into account. So next slide. So for the final question, can we infer the evolutionary history of a gene tree? The answer is yes. We have developed a parsimony method which can accurately infer duplication losses and coalescence. But being parsimony, it does not use branch length and stuff. So next slide. Sorry. So give me a second. So can we make probabilistic inference? So here's an exemplar tree. Now we want to compare 2 histories, 2 more. Yeah. So one is with 1 duplication, one is with 1 duplication and 1 loss. So can we probabilistically distinguish these histories, like maybe looking at the branch length or time of duplication or something like that's what you are trying to do. So, so next slide. So here we plot the probability of generating this tree as a function of time of duplication. The yellow curve is the probability of generating our exemplar tree with 1 duplication, but the blue one is conditioned on 1 duplication and 1 loss. The key takeaway is if the duplication happens before speciation, the tree is unlikely, does not require a loss, but if it happens after speciation, the probability of explaining the tree without a loss becomes 0. This also affects the branch length. Tree from 1 duplication tends to have shorter branch length, but the tree from 1 duplication and 1 loss tends to have very long branch length on average. So by analyzing the branch length, we may get like another clue into the history of the gene tree. Next slide. Okay. So now let's ask, can we infer the expected branch length? Again, we can use our exemplar tree. So if the duplication occurs before speciation, the expected internal branch length is about 1 coalescent unit. But if it happens after speciation, the expected branch length increases linearly more. Yeah, this end of the slide. Yeah. But the probability of that drops, like as you can see, it can drop. So now consider a gene tree with 1 duplication and 1 loss, the end of the slide. Yeah. Yeah. Which is given in blue, which can also occur after speciation, but does not occur before speciation. So if you observe the tree with long internal branching, it is likely that it is coming from 1 duplication and 1 loss. So next slide. So this brings me to the conclusion. The main message is MLMSE is a unified model of duplication, loss, and coalescence. The next one is even a single duplication can create a huge number of gene trees, making inference very hard. Next, it is difficult to infer the history because of just by using the topology alone. And that's it. I would like to thank my advisor, Matt Han, and all the members of Han Lab, and also Joy for helping me. Thank you.
All right. Thank you for that. Because of timing, I'm going to again suggest if you have any questions for Sarthak, put them in the Q&A and we'll be able to move on to our next speaker just because we don't have time to deal with questions right now. However, we've been getting a good number of them coming through the Q&A, and so that's been going pretty well. Spencer, we're going to have you wait just, another 20 or so seconds.
You can give me a little wave or something when you're ready for me and I'll start.
Yep, I can also just tell you when you can start.
That works too.
All right, you can begin whenever you are ready.
All right, thank you so much, Mike. And hi, everybody. I'm Spencer Babernacki. I'm a PhD candidate at Louisiana State University. I'm so excited to be here to tell you all about new insights into the coevolutionary history of pneumocystis and their mammal hosts. And I know I just said the title, and so many of you are wondering what the heck even is pneumocystis, and that's okay. I'm not surprised. This is an unusual genus of yeast-like fungi, and they're obligate parasites of mammal lungs. They're found only in mammal lungs, nowhere else. They're so dependent on them that we cannot even figure out how to grow them in culture. And pneumocystis can cause fungal pneumonia in immunocompromised hosts, but usually they persist at very low levels in the lungs of healthy mammals. Now, as far as we can tell, pneumocystis are found in all mammal species. I even documented pneumocystis in a platypus for the first time. It's one of those weird egg-laying mammals, the monotremes. And pneumocystis fungi are also host-specific. Mammals harbor fungal species that are unique to them, or at least unique to a few closely related species. And it's these two factors that make pneumocystis a fascinating system to study the evolution of parasites and how they colonize different hosts, because we can track their co-phylogenetic history with mammals over tens of millions of years. This lets us understand how events like co-speciation and host switching have influenced their evolution at both recent evolutionary timescales and deep timescales, like when the placentals and the marsupials and the monotremes are all diverging from one another. Now, because of this host specificity that tracks mammal taxonomy, this single genus, Pneumocystis, may be as diverse as all of class Mammalia. That is over 6,000 species that may be out there. But only 6 Pneumocystis species have actually been described. 6 out of 6,000, that's 0.1% of their total biodiversity. Now, this is pretty frustrating because if we haven't documented species in the genus, we can't actually use it to answer those important biological questions about parasite evolution that I mentioned. So if pneumocystis is so interesting, it's so species rich, why do we only have 6 and not 6,000 species described? Now, a major problem is that only a small percentage of mammal diversity has even been screened for pneumocystis. Now, in a paper I published a few years back, I found that it's less than 4%. Remember, we cannot culture pneumocystis. We need lung tissues to study them. But lungs are not commonly collected by museum mammalogists. And even if you do have mammal lungs, typical healthy individuals have extremely low loads of pneumocystis. I'm talking the percentage of pneumocystis DNA can be so low that it would take 100 billion Illumina reads to sequence just a single read from pneumocystis. At most, you might see 0.01%. If you're lucky, that's still only 1 in 10,000 reads. And because of this, most pneumocystis diversity is understood only on the basis of 2 readily amplified mitochondrial loci. These are largely considered unacceptable evidence to describe new species, and they may not be phylogenetically reliable. Now, the good news is that some natural history museums, like us at the LSU Museum of Natural Science and the Museum of Southwestern Biology, have begun to regularly collect lung tissues from mammal specimens. But even if we had lung tissues from every mammal species on Earth, we still face this problem of low fungal loads. So how can we unlock the potential of these museum collections for studying pneumocystis? Is there a way we can sequence more than just these 2 mitochondrial loci? Now, in this quest, the approach that I'm presenting today relies on target capture, where you design probes that match and pull sequences you're interested in out of mixed samples. Domagalska et al. Found that this can isolate genomic pathogen DNA from host samples with about 0.006% or more target DNA. But these folks were interested in a species they had a reference genome for already. I'm interested in novel pneumocystis species. I have no idea what their genome sequences look like, so I decided to identify ultra-conserved elements, or UCEs, from the few pneumocystis genomes that have already been sequenced. Now, we hoped that regions shared by all these sequenced taxa would let us amplify DNA from any pneumocystis species. So we used the program Phylogenetics to identify UCEs from the 7 available Pneumocystis genomes, represented here by pictures of their hosts, as well as 2 closely related species outside of this genus. And we ended up with over 30,000 probes targeting about 2,700 loci throughout the nuclear genomes of these fungi. Great, we designed some probes. We think that they're universal, but will they actually work in practice? So to test them, I worked with 96 samples from a variety of hosts representing these 8 mammal orders, and we knew that they were positive for Pneumocystis based on our previous work with those mitochondrial barcodes. Now, the target enrichment procedure requires pooling samples with similar loads of target DNA, so I estimated Pneumocystis loads using a qPCR protocol. Samples were then pooled and run through a dual round enrichment, which has been shown to improve recovery of target DNA in the case of these low pathogen loads. You basically enrich the DNA, and then you take that product and you enrich it again. So after enriching and next generation sequencing, how many UCEs did we actually recover? Here I have a scatter plot showing the number of recovered UCEs plotted against the amount of Pneumocystis in each sample as measured by the CT value. Now, because of the way that qPCR works, a lower CT value actually means more Pneumocystis. So just to orient you, this was our most heavily infected sample from a macaque with severe pneumonia. It was a reference sample we knew to have about 1% Pneumocystis DNA, and we recovered over 2,000 UCEs from this sample. And this is the sample with the lowest infection load we recovered Pneumocystis UCEs from. This sample has 7 zeros before that 1. That is equivalent to only 1 in 10 billion Pneumocystis cells in this lung. And we recovered over 400 UCEs from the sample. 400 loci from 1 in 10 billion cells being on target. So overall, we find that there is a correlation between Pneumocystis loads and the number of UCEs recovered. More Pneumocystis, more sequence loci. That all makes sense. And we got hundreds to thousands of loci from most of our samples. We would have only collected 2 mitochondrial genes from these samples before. So great, we now have a breakthrough in our capacity to study Pneumocystis. But are these data phylogenetically useful? What do we find when we use them to infer a phylogenetic tree? Now, I wanted to infer a backbone tree for the whole genus Pneumocystis. So I selected 21 samples representative of the host diversity that we successfully enriched. This included Pneumocystis from 7 mammal orders. We had bats, shrews, rodents, pigs, primates, a hare, and even that platypus sample that I mentioned earlier. I then followed a standard phylogenomic pipeline to process our UCE data with some quirks and adaptations due to the unusual nature of this system, but it culminated in the inference of a concatenated species tree in IQ tree and a coalescent tree in Astral. Now, I was very happy to see that with 794 UCE loci, both Astral and IQ tree inferred this, the same largely well-supported tree from these data. Now, other than these samples here that have asterisks, which are from available reference genomes, all the samples in this tree were enriched for this project, the first time that we have these genome-wide nuclear data from Pneumocystis. Now, you can see that all the Pneumocystis samples are colored according to their host order. Now, these deep relationships of Pneumocystis from distantly related hosts were previously extremely uncertain because of limited information in those short mitochondrial barcode loci. And you may have immediately noticed Pneumocystis clustering based on host order. So there does seem to be influence of host identity on recent Pneumocystis relationships, possibly because of significant co-speciation. But those of you familiar with mammal systematics may have noticed some oddities. The deep relationships are completely discordant with mammal systematics. It's kind of chaotic. And zooming in on just one prominent example, these egg-laying monotremes are sister to all other mammals, whereas Pneumocystis from the platypus is found to be sister to Pneumocystis from bats with strong support. So this got us wondering about when Pneumocystis originated. And with these new data from across the diversity of the genus, we are the most equipped we've ever been to estimate its crown age. If Pneumocystis has had a long co-evolutionary history with mammals since their origin, then this deep discordance would be surprising, given that co-speciation is thought to be important. After all, we do see this clustering based on host identity at the tips of the tree. But if Pneumocystis originated after the crown age of Mammalia, host switching would then be necessary to colonize all extant groups of mammals, which would explain their phylogenetic discordance. So I went ahead with estimating the crown age of the genus Pneumocystis. Unfortunately, Pneumocystis and their close relatives are not good leavers of fossils, so I had to pull UCEs from the genomes of taxa across the entire fungal phylum Ascomycota and infer this phylum-level tree, because while here is the subphylum Tephrenomycotina, to which Pneumocystis belongs, the closest relatives that have fossil calibrations are all in this subphylum over here, the Pizizomycotina. So after setting fossil calibrations at these 9 nodes, I estimated divergence times using 2 methods, a penalized likelihood approach in TreePL and a Bayesian approach using MCMC tree. And there's a lot of interesting things in this tree that I found, but I don't have time to talk about them all right now. I want to zoom in on Pneumocystis. Now, the Bayesian MCMC tree estimated an origin of Pneumocystis 98.7 million years ago, right near the beginning of the late Cretaceous. The 95% confidence interval was between 76 and 121 million years. TreePL only gives point estimates, not confidence intervals, but it told us that it was 75.7 million years ago, right at the lower end of those MCMC tree bounds. So these 2 lines of evidence together support an origin of Pneumocystis during the late Cretaceous before the KPG boundary and extinction event. And when I saw these numbers, it actually gave me chills. It was one of those moments where I was reminded how exciting it is to be a scientist. And that's because these dates align with a significant period in mammalian evolutionary history. Therian mammals, that's both placentals and marsupials, are thought to have experienced a major ecomorphological radiation during the late Cretaceous. And it's when placental mammals in particular are thought to have originated and begun to diversify. And let's talk more about this crown age of placental mammals. Similar to this study here, many molecular studies have converged on a late Cretaceous origin of placental mammals. These 3 papers find that placental mammals likely originated between 77.4 and 105 million years ago, and fossil evidence suggests placentals have a maximum age of no older than 131.5 million years. Now, this is a striking degree of overlap with our estimates of the age of Pneumocystis, likely between 99 and 75 million years, and not older than 121 million years. So we find support for a contemporaneous origin of Pneumocystis with placental mammals during the late Cretaceous. Now, this concordant chronology suggests that the diversification of placental mammals opened up a novel expanding niche space that Pneumocystis colonized and then diversified into via significant host switching. And this can explain that discordant topology that we observe. For example, the origin of Pneumocystis in an early Therian or placental mammal would require a later host switch into monotremes. This explains why platypus-associated Pneumocystis is nested within placental-associated Pneumocystis. Now, I really hope you find this all as fascinating as I do, but I always have this sneaking suspicion when I'm giving my talks that people are watching and wondering why they should even care about these obscure fungi. But I like to point out that at least half of all species are parasites. It's thought to be the most common lifestyle on Earth. But I bet if I surveyed everyone in the SSB, parasites would be the minority of species being studied. And this makes sense. Dependence on a host can make parasites very difficult to isolate and study. We've seen that with Pneumocystis. But this leaves parasites disproportionately relegated to dark and unknown branches on the tree of life. And these obligate microscopic parasites, like Pneumocystis, they occur in many clades. Obviously, in the fungi, Pneumocystis is only one of many examples. We find obligate parasites in archaea, in bacteria, viruses, even if you don't quite call them life, and even some simple animals. And there's probably many more we don't even know about, maybe entire clades that are enshrouded on dark branches. Now, my hope is that methods like target capture can help us collect data from some of these elusive parasitic dark taxa. After all, we went from documenting Pneumocystis based on only 2 mitochondrial loci to having a robust backbone tree of the genus based on hundreds to thousands of nuclear loci. We can describe new species. We can start closing the taxonomic gap in this genus. So I hope similar methods can take obligate parasites from largely unknown dark taxa to this, richly documented and described as we illuminate these interesting branches on the tree of life. And with that, I would like to thank everyone listed here, especially the museums that gave me specimens for this project. And I would really like to thank SSB, who funded this project through a Graduate Student Research Award just a few years ago, and now is including me in this symposium. Thank you so much, and thank you all for listening.
So we've got nearly 2 minutes for questions. So again, happy for things to go into the Q&A. Also, anyone wants to raise their hand and we can call on you to ask it verbally. Any one of the people here as co-hosts, also feel free to just unmute yourself if you have a question. All right, no questions forthcoming. Go ahead.
I can answer a quick one. Yeah, I thought that was really cool, and really good from a systematic standpoint of putting in the hard work to still explore these really understudied groups.
Thank you, Becky.
And so I was curious, do you feel like these approaches would be able to help also translate to, kind of trying to tackle these in a lot of other similar systems that may also have these kind of tight co-evolutionary histories with hosts that might also make them complicated?
Absolutely. So Becky, I think that this is applicable to any of these systems. I talked about parasites because I'm interested in parasites, but really any symbionts, microscopic symbionts that are really difficult to isolate from their host. An example that I'm interested in is another group, Microsporidia. They're either these sort of weird fungi or a sister taxon to all of the other fungi. And I think that we have a few reference genomes for them. That's another system I'd be really interested in applying this target capture methodology to. Great question.
All right,
Awesome.
I'm going to have to cut you off here because it's time for our next speaker to go now.
Hello, everyone. I'm EJ, and today I'm going to talk about an important topic in morphological phylogenetics by first defining what a state space is. A state space is simply a space that includes all the possible states of a character. And so if we look into different kinds of phylogenetic data, DNA and RNA, each of them have 4 options. Their state space size is 4. If you link to amino acids, then that number increased to about 20, but still a fixed number. Things, however, get complicated when we look into morphology, such as the inner ear, in that there's hardly any predetermined characters, let alone their state space. And so this is a problem when you try to apply model-based phylogenetic inferences, because most morphological matrices will have many characters with few number of states, such as the binary characters or some 3-state characters, but then there will also be some characters that have many states. And this becomes a problem because by default, the most commonly used maximum likelihood programs that include RexMel, IQ-tree, will treat all of these characters as if they evolve under the same model. And so all the characters shown here will be treated as 6-state characters. But even if we don't think about the programs, there's a difference between the real-life state space, the one you observe based on your data, versus the true state space, the state space that is really the potential of this character. And so this mismatch between the model state space and the true state space is what I call the state space misspecification, or SSM. And I'm interested in how SSM can have an effect on phylogenetic reconstruction, especially regarding the topological precision. I'm also interested in how these different tree parameters and matrix parameters can have an effect on the magnitude of SSM. And so what I did is I designed a lot of scenarios varying these different parameter values. I'll share with you some of the more interesting ones. Here's how the simulation works. For each of the scenario, I generate 50 trees with desired number of taxon. And then for each of these trees, I generate matrices that consist of some binary characters and some multi-state characters. These matrices are then used for phylogenetic reconstruction under either the SSM setup or the SSA setup, where the incorrect setup will treat all the characters as if they are multi-states, whereas the correct setup will treat the binary characters as binary, multi-state characters as multi-states. The reconstructed trees are then compared against the reference tree to assess their similarity, which will be a measurement of the reconstruction precisions. The metric I use is clustering information-based distance, and it works in the following way. You first try to find the most similar split between the two trees, and then you ask the question, how does taxon assignment on one of the tree with respect to that split give you confidence on the taxon assignment on the other tree with respect to the respective splits? And if you ask the question, enumerate through the splits, you will get an overall measurement of similarity where the opposite will be the distance. And so here's how I will present the simulation results. I'll first ask, how does the parameter values affect the overall tree distance? And then how the two setup perform relative to each other. And finally, how the parameters have an effect on the relative performance of the two setups. So first, looking at state space size difference, what I note is that as state space size increases, the overall distance decreases. This makes sense because when you have larger state space size, it means that the data is less likely to get saturated, and so you have more information. As you can see here, the blue circles, the SSA, the correct setup, is outperforming the incorrect setup, the SSM setup. And that difference actually increases as state space size increase. Moving forward, looking at the proportion of binary characters and mutation rate, there's an overall increase of tree distance as mutation rate increase, which again makes sense because higher mutation rate, higher possibility of saturation. SSA is again outperforming the incorrect SSM setup, although there's not really much difference in the relative performance as mutation rate increases. When increased to 50% binary characters, the overall patterns remain largely the same, except that SSA outperform SSM even more, and that there's also a slight increase in the relative performance as the mutation rate increases. However, things get really interesting, if not confusing, when further increase the binary character proportion to 90%, in that while mutation rate is low to moderate, the overall pattern is pretty much the same as the previous simulations. When mutation rate is high, all of a sudden, the incorrect setup, the SSM setup, is outperforming the SSA setup by having shorter distance. This is particularly important because this is where most morphological data sets belong to when there's high mutation rate, high proportion of binary characters. And these patterns become even more obvious when I did what I call the false space simulation, where all the characters, or 100%, are binary, but they are treated as multi-states under the incorrect setup, in that the overall pattern is same as the 90%, except that the mutation rate that result in this counterintuitive effect actually start at an even lower rate. So how could this be possible? How could it be that an incorrect setup is giving you the better results than the very model that is actually used to generate the data? Well, actually, this is perhaps not that surprising, given that almost 30 years ago, some of the big figures in phylogenetic methods have already noted that maximum likelihood can actually produce biased results because phylogenetic inferences include topological changes, and that these changes lead to parameter space change. And so maximum likelihood estimation does not enjoy the statistical consistency it has when estimating single parameter values. But this only explains the plausibility of the results. What about the actual cause of the results? It took me a very long time to kind of figure out what is going on. So here's the thing. Binary characters are more likely to get saturated, especially when there's high mutation rate. This means overall weaker phylogenetic signal. And this is where SSM comes into play. By modeling a larger state space, it introduced greater punishment for homoplasy. And I'll show you what I mean by that. Here are two character pattern hypotheses, one of them the homologous one, the other the homoplastic one. Now you can calculate the likelihood under a two-state model or a seven-state model. Now in both cases, the homologous pattern, the hypotheses on the left, have a better likelihood. But what I want you to note is that tree with homoplasy is much less favored, as you can see from the difference of the likelihood. What this translates to is that if the other characters together do not show strong preference to either of the trees, then merely based on how you model the character's state space, you can end up preferring different tree hypotheses. And so if we assume that the correct tree has more of these homologous patterns, then the overall effect of SSM is that it will bias tree with more inferred homology, and in this case, the true tree. So how would this bias show up in the data? Well, using another simulation with 4 taxon, here you have these topology informative pattern. Essentially, you have the A having the same states as B, or C, or D, where AB will be the homologous pattern. I again did this false space simulation, so the characters are binary, but treated as either 2-state, 4-state, or 10 states. Now, if we think about the relative abundance of the AB, CD, the homologous pattern, when most of the character is like this, which is actually pretty common, then both setups are likely to give you the correct answer. Whereas if it's the opposite, then both are likely to get it wrong. However, during the borderline cases, when it is slightly more than 0.5 or less, where it is essentially an ambiguous situation, then if the two setups disagree with each other, then if the bias that I just described is true, then you will expect that the wrong setup will be the correct one precisely when there's slightly more of these homologous patterns, the A, B, C, D pattern. And this is exactly what the simulation shows, that under a false space simulations, you have this SSM correct SSM ROM scenario when there's slightly more AB than the AC or AD. And this pattern actually becomes even more obvious when further increase that discrepancy to a 10-state false space simulation. And simply because there's slightly more of this AB pattern, the SSM outperform SSA. And so given this bias to a tree with more inferred homology that actually helps in this case, does this mean that we should use the wrong setup? Well, in my opinion, it is at least worth for assessing topological stability. However, there's an important caveat, which is that phylogeny is a combination of topology and branch length. So what this means is that when branch length is taken into consideration using another metric that take into account branch length, then SSA is actually doing much better relatively. And this has also been noted in some of the very recent studies in Bayesian inferences. In fact, you might just have heard of one of those. So thus far, I only talk about maximum likelihood. What about maximum parsimony, which is still very commonly used in paleontology? Well, I decided to test it by using parsimony with the simulations that I have, and I noticed a striking similarity between the parsimony tree and the SSM tree in that they are even closer to each other than to the reference tree. This is maybe not that surprising given the simulations I just shown, because parsimony will strictly choose the tree with the most common ABCD pattern. And so there is a significant overlap of what SSM and parsimony prefer. However, if you want a more general explanation that also take into consideration more complicated scenarios, then we have to look into this TS97 no common maximum model that someone actually just brought up in the QA. So the TS97 models guarantees that if you minimize the total steps, which is the parsimony score, you will also be maximizing the likelihood, and so you will get the same trees. But the problem is that there is a caveat here that the state space size is assumed to be fixed. And so if we introduce a variation there, then this derivation no longer holds. And so we need to make some adjustment. As it turns out, through some numerical exchange, you can make it a product of the exponential of weighted steps multiplied by a constant. What this means is that if you minimize this instead, which is weighted your characters with the log of the state space size of each respective characters, you get this weighted parsimony score that if you now minimize that, you will also maximize the likelihood under the NCM model, but this time taking into consideration the state space variation. So given this, does SSA weighting actually improve maximum parsimony? Well, I don't have the no common maximum model data just yet. However, I do have those that was generated under the MK model for the other simulations. And it does shows that the weighted maximum parsimony is outperforming the maximum parsimony, the unweighted version, by having shorter distance. And so the overall takeaway from this study is that state space misspecification is a problem, and it's not just about underestimating branch length, they also reduce topological precision. Although just to make things complicated, there's this important exceptions when you have high mutation rate and high proportion of binary characters that you will actually get better topological precision. I also showed that parsimony is perhaps not as truly model-free as one would have thought, and that there is this close connection of the unweighted version of the parsimony with the state space misspecification. And so if you want to acknowledge the state space variation, you'll want to consider partition your data sets if you are doing a model-based approach, or weight your character if you're doing a parsimony approach. And so to facilitate this, I've created this simple tool, Morphopars, which will do precisely that. It can partition your data sets or weight your character. It also includes some of the different format conversion and handling polymorphic encoding just to facilitate morphological phylogenetic analysis, because some of these measures can be quite messy. And here's the motif in case you are interested. So with that, I'd like to thank all the people who have provided valuable comments to me, especially my PhD committee and my PI, Dr. Beaver. And I encourage you to check out Morphopars. Thank you.
All right, so we should have time for 1 or 2 questions. All right, April, go ahead.
Hi, EJ. Very nice talk. I was curious. So, Cockerell 2024, and then Jen Hoyle-Cuthell wrote back in 2015, kind of a big simulation-based exploration of this issue in Parsimony. They both found very strong branch length effects here, which I think gets at the issue of homoplasy. Did you find similar issues where if you're not working with long branch attraction trees, this problem is lesser, or did you find, what did you find?
Yeah, so I didn't test specifically long branch attraction thing, but actually I did. So when I was doing the simulation showing the bias, that bias will be reverted if you have like these purposely designed long branch trees, which is again because of this bias that if you have this bias, the wrong setup helps you. But if it is the long branch trees, then because these trees are skewed from like the normal tree that you expected, then this time that bias actually hurts the incorrect setup.
Okay.
Yeah. But most of the trees are like very generally, like it's a general pure burst trees. And so there's not really like clear indication of like the long branch attraction issue. And I haven't specific test.
All right, I'm gonna have to cut you off here because we need to transition to our final speaker of the session. And you are free to start presenting now.
Yes. Can you see my screen?
Yes, we can.
Okay. So hi, everyone. This is Junman. I'm a PhD student at Southern Illinois University. I want to start by thanking the organizer for the opportunity to share my work today. And most of what I'll be presenting comes from my master research. And it focuses on developing a new matrix-based filtering method to help resolve a long-standing controversies in the phylogeny of Chondrichthyes. So over the past couple of decades, the cost of the DNA sequencing has dropped dramatically. And as a result, more and more species has been sequenced and assembled to high quality genome. And this has brought the phylogenetic research into the phylogenomic era, where we no longer limited to a few genes. Now we can analyze hundreds or thousands of loci. So for instance, more loci typically will reduce the stochastic error, and think of how the bootstrap value often reach 100% and stabilize as we add more data. But more data doesn't mean better or more accurate result. So phylogenomics also introduced new challenges. So different genes may have different evolutionary history, and this leads to the gene tree discordance. And some of the discordance is due to biological sources, such as the incomplete lineage sortings. But another major source is analytical systematic error. So for example, using the wrong model, ignoring the base compositional bias, etc. So to address these kind of issues, particular analytical bias, two main strategies has emerged, the metric-based filtering and topological-based filtering. So metric-based filtering looks at general signal across the genes, such as the GC content. And these metrics help identify the potential misleading genes, so which can be filtered out. And topological-based filtering takes a different approach. It compares how well each gene supports to the tree topology and to identify which genes best support a given phylogenetic tree. But both approaches have major limitations. So metric-based methods evaluate the global signal across the entire genes. They often ignore the localized problem, like just a few sites or textures, and they can mislead the phylogenetic inference. And on the other hand, the topology-based methods require the user to provide the candidate topologies that create a circular problem, because it is too subjective to choose which tree are potentially true for statistical comparisons. And this brings me to the specific case of Chondrichthyes. At the order level, their monophyly is well established, same as each major supergroup, superorder, and like the Galeomorphy, Scholomorphy, and Bacteroidea. However, when we zoom out on the relationship among this group, especially within the Islamo branch, things get messy. So traditionally, most research have played the Galeomorphy and Scholomorphy together as the sister group, with the Bacteroid forming a separate clade. But depending on whether study use mitochondrial genes, nuclear genes, or morphological trait, the result varies. Some even suggest the Bacteroid is more closely related to the Galeomorphies, and in this case, hexcentrifugal fall outside the Scholomorphies, and that's the unexpected pattern we saw in our initial result as well. And here is one of our early results using a phylogenomic dataset without filtering. And you'll notice that the hexacentriformes appear very close to the outgroup chimeriformes with an unusually short branch length. And that's a red flag for a potential systematic error. Interestingly, the relationship within each superorder looks fine, but between the superorder, they look suspicious. And this suggests to us that localized signal were driving the problem. So we asked two key questions. Are systematic error biasing the Chondrichthyes phylogenomic result? If so, can we correct them? And how does our new metric-based filtering approach compare with the traditional one? So to explore this, we use the Evo marker pipeline to design a base for Chondrichthyes using a single coding sequence from 6 reference genomes, and that was back in 2019. And we end up identifying 24,000 candidate markers. Except for gene capture or target enrichment data, we also compile data from public database like transcriptome RNA-seq data, whole genome resequencing data, and genome assembly. For taxon sampling, we collect at least one species per order in order to explore the Chondrichthyes phylogeny at the ordinal levels. We process the data through a detailed pipeline. First, we filter and align the sequence based on missing data and taxon coverage, ensuring that each gene remain monophyletic at ordinal levels. We then prepare several versions of the database, a full nuclear dataset, and a first and second codon precision dataset, and amino acid datasets. To explore whether the outgroup attraction was twisting the tree topology, we also prepare 2 more datasets. One that added more outgroup to mitigate the long branch between the chimeriform and the elastomer branch, and another that removed the hexacentriform entirely. So all the datasets were analyzed using maximum likelihood in IQ3, and we're testing both traditional and novel filtering methods using the top 300 most informative loci. We also explored the topology-based filtering using GLS. So from the unfiltered data, we saw the same controversial result as the previous study. The bacteria was recovered as assistant to the galeomorphs, and we also calculated gene concordant vectors and find that the red nodes had a very low value, which matches the known conflict in the Chondrichthyes phylogeny. When we compare the result across the dataset, we saw some consistency in the bacterial topology. However, the elastomer branch showed 2 conflicting patterns, and only dataset without the hexacentriform supports the elastomer branch type 1 topology. And we define this as a hypothetical topology 1 and 2, and use them as a hypothesis to do the GLS filtering. The sum of the delta GLS value is strongly favor type 2 topology, even after filtering out the outlier based on the scatter plot and reconstruct the tree. And this is because these loci have balancing phylogenetic signal and not extreme value. But again, this method is critical reasoning and assuming the truth of one tree to test the other, and which doesn't sit well with us. So that's why we focus instead on developing a better matrix-based approach. So traditional metrics like the RCFV only capture the whole gene signal. They miss the important difference between the taxons. For example, this true gene has the same overall GC content, but very different GC distribution across group. But the RCFV of these genes are the same and fail to detect the group variation. So we introduced 2 novel metrics, the GC difference based on the absolute difference in GC content between group, and the absolute branch length ratio based on the relative branch length between the major lineage. And we designate the chimeriformes as our group, hexacentriform as the in-group 1, and bacteria plus the galeomorph as in-group 2 to investigate their group variations. And here's the example. The 2 genes have similar global GC content, but only one shows a significant difference between the group. And our GC different metrics pick that up, and while the traditional metrics fail to do that. Similarly, our branch length ratio captured the lineage-specific difference that could indicate the systematic bias. And when we apply this new filter, we find something striking. One of the novel methods, the branch length ratio matches, recover the elastomer branch type 1 topology, which is widely accepted in the mitochondrial study. While the absolute logistic difference matches still fail, it could be that the branch length offer more information, not just logistic content. We also use the same dataset to conduct coalescence analysis, and novel filters show positive results that support shock and ray are monophyletic. We're also testing the relationship among all of the matches using the Pearson correlation analysis. The color of the circle represents the direction of the correlation. Red means negative correlation, and blue means positive correlation. And the circle side refers to the correlation coefficients. Only a significantly correlated pairwise group will be labeled with an R value. So as expected, the traditional matches were highly correlated with each other. But our branch length ratio matches was highly independent, except for a weak correlation with the evolutionary rate. So which makes sense because both involve using the branch length for calculation. Finally, we look at how much overlap exists among the top 300 loci selected by each method. So most loci chosen by the novel matches were unique. About 80% of them didn't overlap with loci chosen by the traditional filters. In contrast, the traditional methods share a lot of loci and often gave us the same problematic result. To write up, our study showed that even high support phylogeny can be misleading if systematic error remain unresolved, and the deep branch relationship within the Chondrichthyes is vulnerable to this bias, and our novel metrics, especially the branch length ratio, offer a new and more targeting way to detect and remove the misleading loci by focusing on local variation rather than the global average values. And this gives us a useful filtering step before we begin to build a tree. And interestingly, traditional matches tend to be strongly correlated with each other, but our branch length ratios stand apart, giving us an independent signal to work with. In the future, the Chondrichthyes base can be refined and improve gene capture efficiencies, and this marker can also be used in population genomic study. More broadly, our filtering methods may help resolve other difficult phylogenies, especially in the clay with unusually long or short branches. And if you're facing similar challenges in your own phylogenomic study and are interested in testing these new methods, so please feel free to reach out. And here's my email. So finally, I want to thank my advisor, LabMate, collaborator, and our funding sources. And again, I appreciate the Evolution meeting organizer for making this event possible. I'm happy to take any question. Thank you for listening.
All right, and we have plenty of time for a couple of questions. So either into the Q&A if you'd prefer to write them out, or feel free to raise your hand and we will let you unmute and ask them verbally. All right, so we had something show up in the Q&A, which is asking if the hexanchiformes have a long branch length.
So in our result is the hexanchiforme actually has a very short branch length. So that's why it closer to the outgroup, the chimeriformes. So we're thinking about it's also like the long branch attraction, but this time it's not a long branch extraction because the outgroup is so short, and the hexanchiform is also short, have a short branch length.
All right, I will ask. So what are you planning on extending this into next?
So since the methodology for this paper is we developed probably 24,000 of the gene capture base. So but none of them, most of them actually has a very low gene capture coefficient efficiency. So the next step is we try to, based on the current data, to filter out which data they have a higher efficiencies to do the gene capture. And because we developed this kind of new filtering methods, so we try to apply this on other study. because we think in other taxon or other species, they probably also have this same of the issues. So we try to test it.
Well, if we have no other questions, we'd like to thank everyone for their contributions here. As always, I am happy to be in the Student Award Talks. I'm very happy to not have to judge them, because that is going to be a lot more work for the panel of judges. So we're going to stop the recording here. Let's see.